Okay, so let's get started. Hello, everyone, and welcome. Thank you for joining us today for our exclusive interactive session. I will be your host today for the special event. We're here to dive into the groundbreaking research published in Nature Medicine. It's titled T and B cells responses against Epstein-Barr Virus and PSC. So this study uncovers a strong immunological link between Epstein-Barr virus, EBV, and PSC. So this insight can open up new avenues for understanding, diagnosing, and treating this complex disease. I am excited to introduce Hisham from Keele University, who is the primary author of the study, as well as Stan, the CEO of My Laboratories, who will be doing the demo on how to reproduce the study and the publication without any coding. So for today's session, feel free to drop any questions you have in the chat, and we'll also answer them at the end. We also have Dimitri Bulletin here, who is the CTO of My Laboratories, who can also help explain things further. So with that, let's get started, and I'm excited to introduce Hisham.
Yes, thank you very much for the kind introduction, and it's really a great pleasure to be here. So I'll just share my slides. I hope you can share, you can see it. You can see my slides, right? Okay. So just feel free to interrupt me in the middle. I mean, it's not a problem. I mean, we can just discuss things on the spot. Okay, so today's talk would be about how can we draw a mechanistic insights about immune-mediated inflammatory diseases using immunogenetics methods. And I would like to start first by giving you a little bit of a model of how we think about the role of adaptive immune system in diseases. So the world is complex, and we need to simplify it a little bit to a way that we can interpret and draw hypotheses, and then we can add complexity when we need. For the rest of this talk, we are going to assume that innate immunity doesn't exist in a way, and it's only adaptive immunity. And we are just going to focus on the specific properties of adaptive immunity, namely that once we start to expose to an antigens, and if we formulate an immune response to it, then we are going to have an immune memory to it. This immune memory is going to be like a fingerprint to this particular antigenic exposure. The properties of this antigenic exposure is like three kind of layers. You start to have a unique VDJ receptor, such as a unique T cell receptor, a unique B cell receptor that would recognize part or antigens or epitopes coming from this primary exposure. You would have a specific transcriptional program that will be present in the site. This T and B cells, for example, thinking about T helper 1, T helper 17, T helper follicular helper. And there'll also be some sort of epigenetic memory that would fix and maintain this kind of program. For the rest of the talk, we are just going to focus into this kind of unique aspect of having a specific receptor recognizing part of this antigenic exposure. So we can think of the immune repertoire that we are analyzing and dealing as a collection of antigenic exposure, assuming that naive repertoire is not very interesting, and then that's like the effector part is also not the most important part. It's mainly the memory aspect of it. So when we try to look at the immune exposure trajectories or history, we are going to see that after we are born, starting from the first trimester where we start to form like the very primitive T and B cells, and then after we are born, we get colonized with the microbiome, we get exposed to an antigen, there is a series of antigenic exposure that will be included in these arrows here. And from this antigenic exposure, we start forming immune memory, and this immune memory is going to be long-lived. Of course, here we make a simplification that this line doesn't fade and that we don't get like reinfection, but we can just focus on the main aspect that we have a series of antigenic exposure that result in the formation of specific immune memories. And when we analyze the repertoire, we get to analyze at least a subset of these antigenic exposure memories. However, to make this like model complete, we need to assume that there is a lot of genetic variability that we start with. We start from a specific genetic combination in terms of genes, germline variation present within the V and J genes, and as well as within the HLA region. And this is a fixed immunogenetic effect that would have its exist regardless of the antigenic exposure history. So the immune repertoire, when you think about it, is a combination of these fixed immunogenetic effect that inherited genetically, and then environmental antigenic exposures and interaction between both of them. So let's take one step back and think about these fixed effects that come from the HLA. What are the HLAs in general? So the HLA proteins are the human version of the major histocompatibility complex, the MHC. We have two classes. We have class 1 and class 2. Class 1 present peptide toward CD8 T cells, and class 2 present peptide toward CD4 T cells. The problem or the beauty of the HLA system is they are extremely diverse. They are located within chromosome 6 and the petite arm, and then we have class 1, 2, and 3 regions focusing on 1 and 2. There is thousands of HLA class 1 alleles and thousands of HLA class 2 alleles. So there is a high degree of polymorphism. And why should we care about this polymorphisms? This has to do with the genome-wide association studies. So there is a lot of diseases called immune-mediated inflammatory diseases or autoimmune diseases or chronic inflammatory diseases. I mean, you can name it. It's up to interpretation. But the whole fame of these diseases is there is inflammation. This inflammation would lead directly or indirectly to tissue destruction and then tissue remodeling. And when we did genome-wide association studies to try to see, comparing cases to controls, where the variation or where the genetic part of this disease might be coming from, we usually see something like this. We see that the very strong peak present in chromosome 6, people call it like Manhattan plot. You can even call it a Dubai plot now, because you have this kind of one big peak present in chromosome 6. And this one long peak present in there usually indicate it's resolved to the HLA region, so the MHC regions. That means these proteins that present peptide to HLA are implicated or associated with the disease, and the B values can be insanely high. There is also usually a lot of other like SNPs or other polymorphism or genetic association studies, but in other non-HLA regions present in there. But it's usually in these diseases, there is this high peak coming from the HLA region. So in most of the autoimmune diseases, or in most of the immune-mediated inflammatory diseases or chronic inflammatory diseases, we have the genetic signals. We know that the HLA is implicated, but we don't know what is the antigens that's causing these diseases, except one disease, which is celiac disease. So in celiac disease, although it's not 100% clear, but we know that the exact antigen doing this is gluten, which is coming from bread, actually. So it's a gluten present in bread proteins. So the whole idea is, if you look at the GWAS of a disease like primary sclerosing cholangitis, and you look at the GWAS of a disease like celiac disease, you see that both of them are really similar. But in celiac disease, we know what antigen triggers or causes this disease, or I mean, causation is very hard to prove, but at least is very responsible for the pathophysiology of this disease. However, for these diseases like PSC, we wouldn't know what causes them. So we want to understand what are the antigen that drive these diseases. And you want to do this using an immunogenetic approach. This was like accumulated into this paper titled TMB cell responses against Epstein-Barr virus and primary sclerosing cholangitis. So let's dig into the paper and try to basically, like, I will guide through how we basically arrived to Epstein-Barr virus, because it was at least a fun journey. First, before we jump into the details, I think primary sclerosing cholangitis is not a very famous disease, so I would like to give you an overview about it. It's a very rare disease. What happened is that there is inflammation that happened in the bile duct. It's not in the liver, it's in the bile duct. And there is an inflammation, there is then fibrosis, and this fibrosis causes obstruction in the flow of bile. So the bile, which is cytotoxic, that basically contain a lot of waste product, cannot leave cannot basically flow normally. So it's like being clogged. And when it goes back, it starts to build up in the liver, and which is basically cytotoxic. It basically causes like liver failure on the long term. It can also cause cholangiocarcinoma, which is a very bad form of like cancer that happened in the liver. It's also associated with an IBD-like phenotype. Basically, there is an inflammation in the colon. So it's like ileal backwash, there is an inflammation in the colon happen, and basically no one knows why. Sadly, for PSC, there is no treatment. You would basically try to clinically control the patient state, and up to the point where they need a new liver transplantation. And even after liver transplantation, you might have a disease recurrence. So it's a very devastating disease, and we don't know why some people get the disease and others not. We know from the GWAS that the HLA region is strictly implicated in it, or strongly implicated in it, because if you look at this kind of a GWAS plot, Manhattan plot, you see the HLA region is highly enriched in there. We also map this region to know which HLA allele would be implicated in it. So for example, we have the ancestor haplotype present in there, and then we know that specific HLA allele are strongly enriched in PSC. These include the HLA-BO801, the HLA-DRB30101, and the HLA-DRB10301. These form what's known as the ancestral haplotype 8.1, and it's strongly enriched in individuals with PSC. So given that we know the HLA allele, we wanted to understand further what are the antigen and what are the T cells that are like expanded, implicated in it. So what we did here, we assembled a large cohort of primary sclerosing cholangitis patient and healthy control. And then we profiled the T cell repertoire of these individuals, specifically the T receptor beta. I mean, I don't need to explain this to such audience, but basically we have this kind of memory T cell that will be present. We didn't sort, but if focusing on public clonotype would be mostly memory. And then we have the specific VDJ receptor. We form primers again as the V and the J, and then we can amplify the region. We end up with a repertoire that will be present. Once we have this repertoire, then we wanted to identify which one of them is statistically implicated or associated with the disease. So what we did, we basically used a one-side Fisher exact test to identify the clonotype that would be associated with PSC, and then we choose the p-value based upon a permutation-based false discovery rate. So we identified the clonotype, which was 136 in the beginning, then we clustered them to identify similar clonotype, ending up with around 1008 clonotype. And then we try to look how the burden of these looks like. So of course, because they are associated with PSC, each individual that have PSC have more of these clonotypes relative to healthy controls. So, okay, so this was our discovery cohort, and we wanted to be more like strict at what we're seeing is really as a reproducible strong signal that would be present across cohort, or maybe a statistical artifact of this cohort of the properties of the method. So we looked at two other cohorts. One of them was coming from the United States, which contained patients that have PSC and not PSC. They have a UC or IBD. And we see that the clonotype were present like at much higher expansion level in individuals that have PSC relative to patient without PSC. We also repeated this in a Norwegian cohort containing individuals PSC and individuals without PSC or healthy controls, and we could see that the expansion was higher. So we know that these clonotype might be a reproducible finding. They are present in different cohorts, and then we can identify them across geographies and technologies. So, but are they present in the liver? Because we identified them from the blood. Here we used previously published data sets. One of them was Eliakoza et al., where we tried to see the overlap with what was published, and we found that some samples has a lot of these clonotype, other sample had few. Then we took a second data set that contained the blood and the liver. One of them was coming from a different data set, Hendrickson et al., and these are coming from the same patient. And we found that, well, there is more clonotypes present in the blood relative to the liver, but they are present. However, the repertoire size, the number of clonotype present in the blood samples was much higher than the liver. And after we normalized, we found that they are quite expanded in liver samples. In some individuals, we couldn't detect them, but in most of the PSC samples, we could identify them. So we're kind of identify what would be the factor that can make one person have a higher burden of clonotype, other burden have fewer. We tried to test everything. It was not age, it was not sex, it was not disease status, it was not a lot of things. It was actually genetics. So when we think about a disease-associated clonotype, usually these are conventional T cells. That means they are restricted toward a specific HLA allele. And as a result, we found that the factor that explain big part of this variability is HLA background of the individual. So here we have the two HLA alleles, the HLA-BO801 and the DRB0301, the two enriched in PSC. And then we try to see, is the signal we see, is it only because of the HLA effect or is it because of the disease? So we have now this kind of two things. We have the carrier and non-carrier, and then we try to compare the expansion between the carriers that have PSC relative to the carrier that doesn't have the PSC, and here we observed like a higher expansion. We also found that the non-carrier that have PSC has it higher than the non-carrier. However, the carriership in itself increased expansion of these clonotype because they are restricted to it. So we know that these clonotype are restricted. I mean, they are conventional and they are restricted to common HLA alleles, mainly here the BO8 and the DRB0301. Then we try to expand this further to try to understand, giving this 1008.
Sorry, Hisham.
Yes.
Apologies. You said that we can interact and,
sure.
and ask questions. I have one question. Can you explain a little bit more details? What is the difference between carrier and non-carrier? What is carrier and non-carrier?
Yeah, so carrier mean individual that have in their gene, they have the HLA-BO801 in their genes. So they are HLA-BO801 positive. Non-carrier, they don't have this allele. So they don't have this HLA. And then we try to compare between carriers that have PSC. So everyone here is HLA-B08 positive. Some of them have PSC, the other don't have PSC. Here, everyone is HLA-B08 negative. That means they have other alleles, like 702 or B08 or 401, whatever. But then we try to compare the expansion between them. And we found that the expansion was higher even on these, like in PSC non-carrier relative to healthy control non-carrier.
Thank you.
Anytime. Then we wanted to expand this further to try to understand toward which HLA allele these clonotype might be restricted. So what you see in this network, each dot here is a particular clonotype. And two clonotypes are connected if they have the same V, the same J genes, and there is only one amino acid difference in their CDR3 region. The coloring of each dot is based upon toward which HLA allele these are restricted to. I mean, maybe in a different talk, we can talk about how can we resolve this HLA TCR associations, but I mean, we can just keep it simple now that we resolve this association for big fraction of these clonotypes. And then each color corresponds toward which HLA allele these clonotype were restricted. And as you can see here, we have like the biggest cluster that we identified were restricted toward what we call the risk HLA allele, for example, the HLA-B0801 or the HLA-DRB0301 or the HLA-DRB1301. So what we learned until now is we identified a specific clonotype that are expanded in PSC and they coming from conventional T cell that are restricted or specific toward risk HLA allele. So what can we do it further from here? We wanted to see if the signals we identified is specific to PSC or to inflammation in general. So we compared the PSC toward ulcerative colitis and Crohn's disease. These are two other immune-mediated diseases that happen in the gut. And most of the patients that have PSC have an IBD-like disease. So that was the justification of choosing these two. And we found that the expansion present in PSC was higher than UC and higher than control, and of course, higher than healthy control. So we know that it's a specific signal of PSC, it's reproducible, we can see it across different cohort, and at the same time, it doesn't seem to be inflammation specific, it seems to be PSC specific. So the question becomes, which antigens are being targeted by these clonotypes? And basically, we were in this stage at around mid-2023, something around this time, and we wanted to identify toward which antigen. So the easiest option for us was let's look at public data sets and see if these clonotypes, which are public by definitions, have been identified in there and toward which antigen would they bind. We found that some were bind to CMV, and also this public data set has a little bit of issue because sometimes you have the clonotype being annotated toward multiple organisms. So we just focus on the one that mapped once, and then we basically try to come up with an enrichment score that would be similar to gene set enrichment analysis. And we found that the clonotype seems to be targeting to some extent Epstein-Barr virus. Then we collaborated with Adaptive, and then we searched an internal data set of Mera-generated assays. So we have a data set of clonotypes and the antigen they are restricted to. And we found that most of these clonotypes are targeting the BZLF1 protein, which is a specific part of the BZLF1, which is an immunodominant part of EBV. And this protein will become interesting because it's a lytic phase protein of Epstein-Barr virus. So we know that this protein, at least a subset of them, is targeting Epstein-Barr virus protein. Okay, so what can we do? Is there a better way to dig deeper into this? So what we did, we took the PSC, compared it to Crohn's disease, and then we did the same procedure again. We found that there is the EBV antigen present in there. We compared the PSC relative to UC. We got the PSC specific clonotypes, and they were enriched for EBV. And we also we identified the T-cell signature that maybe subset of these clonotypes are restricted to EBV. But can we see anything from any other layer of immune information, from immunogenetic information? And here we switch it completely from the T cells toward the B cells. And we didn't do a B cell sequencing, we did it completely different. So the whole idea, again, we know that the exposure would result in the formation of a memory B cell. This memory B cells would generate an antibody. This antibody can live for decades in our body. And when we take a serum samples, we usually get a collection of antibodies that summarize what we have exposed to before. So it's like a white color, and we want to disentangle this into the individual color present in it. So we want to prism it sort of. And the technology to do this, or one technology to do this, there is a lot, but one of them is phage immunoprecipitation sequencing, which has slightly different technologies relative to TCR and BCR sequencing. You take the serum, which contain antibodies, you design a library of antigen that you are interested in. Here we had close to 400,000 antigens. You clone it into a phage, and then you would have the phage expressing the antigen on its surface. You have the phage and it's expressing antigens. You incubate it with the antibodies, and then you pull down the antibodies. If the antibody bind to the phage, it will be pulled down. If the antibody didn't bind to the phage, the phage would just be washed away because there is no antibody binded. Then you can do a lot, you can do sequencing, and then you can do a lot of statistical analysis to identify the antigen bind by the antibodies present in the sera. So you you don't get the sequence of the immune receptor, you just get the antigen itself. So we did this in 202 healthy controls. We did also in 120 patients, and we did the PSC to try to identify the antigen that will be bind. And we found that the number of antigen was different between the control and PSC because there is like a more focused immune response happening in PSC relative to control. However, something that, of course, like people working T cells know that most of the immune receptors, most of the immune responses we have are private. So they are only detected in one individual, and this is a log scale. So you can appreciate that it's a very small number of antigen or antigenic exposure that we can publicly analyze. So there was only 23,000 antigen out of the close to 400,000 that were present that generate immune response in PSC patient and control. We can just jump a little bit further because of the details, but then we compared the public, what we call public antigenic exposure in cases and control, and we found again that there was an EBV signals present in there. So there was multiple of these like dots shown in here were coming from Epstein-Barr virus. There was also something coming from the Clostridialis and self-protein, but we focus on the Epstein-Barr virus because of the T cell responses that we had before. So we observed also from the B cell that there is a higher, let's call it titer of anti-EBV responses. And when we look at the fraction of EBV responses present in PSC relative to healthy controls, we found that they were also higher. So this gives us more weight than there is more EBV responses coming from PSC relative to control. We tried to see which part of the protein would be targeted more often by these immune responses, and it was coming from a protein called BMRF1. And BMRF1, also known as the early antigen diffuse, it's also a second indicator or a protein that's used to identify EBV reactivation or a recent infection. So we wanted to extend this further, and we looked at all the EBV protein, a representative sample of EBV protein. That's not all the EBV protein. And then you can see in the first row on top, we observed an increase in anti-BFRF3, which is known as the viral capsid antigens. And there is also an increase in the EBNA1 protein. So the conclusion of having A and B, and then also E with the BMRF1, which is indicate a lytic phase, that we basically have an EBV reactivation. It's not a new infection, it's just a dysregulated immune response to our Epstein-Barr virus, or there is an EBV reactivation. And this has been a repeated pattern in most immune-mediated disease, rheumatoid arthritis, multiple sclerosis, lupus disease. So that has been basically the main finding. I think I'm almost done with my talk, so I'll just give you like a brief summary that the PSC as a disease is strongly associated with an HLA, and specifically to the ancestor haplotype 8.1. We identified a specific clonotype that are conventional in nature, restricted to these two alleles and associated with it, and that using different techniques like TCR-seq and PSC to look at the immune system from different angles or different perspectives, we observe this kind of increase in anti-EBV responses in PSC relative to controls. So the question becomes now is, can we extend this framework to other diseases? Would it only work in PSC, or can we try it with other diseases? And I just want to show you very, very brief results. We also repeated the same approach in Crohn's disease. We found the clonotypes, and we were also able to validate them. So this kind of highlight that using this population scale TCR sequencing and comparison, you can really pinpoint specific clonotype associated with diseases. With that, I reached the end of my talk. This was a massive work. There is a lot of people also not in this picture that contributed to this. So especially Andra and Aya, and of course the funding agency, because this was this was a bit expensive. And of course, you for your attention, and I'm really happy to take any questions. Thank you very much.
Thanks, Hisham. So before I pass over to Stan, we just have two questions. One is from Dia. She asks, were there overlapping TCRs between the two carrier HLAs?
Do you mean, let me just make sure that I understood the question correctly. Do you mean some clonotype that were associated for the HLA-BO801 and the HLA-DRB-O301? right?
I assume so, yes.
Yeah, I mean, yes, that was that was a big problem. So the HLA-B0801 and DRB0301 are present in the same haplotype. So when you do it statistically, you would sometimes have clonotype that are associated with the entire haplotype. And the only way to resolve this association is to have a sample, to have a large sample, that's number one, and at the same time to have a large population where you have seen, like where the haplotype is not completely preserved, like it's like, it has been recombined. So in our cases, our big problem was not with HLA-B and DRB0301. That was not the big problem. The big problem was with HLA-DRB0301 and with HLA-DQ. There was a handful of clonotypes that we couldn't really resolve or disentangle whether they're coming from the DRB1 or for the HLA-DQ. So that, and at some point it was like, okay, so it's either DRB1 or HLA-DQ. We can't really tell because of this, they are in the same haplotype structure. So if we repeated this was like 10,000, 15,000 individual, then we might, of course, from a different population, then we'll be able to resolve this. But for the time being, within the HLA class 2 allele, that was the main headache.
Great, thanks. And Kevin also asks, how do you determine that the clonotypes are specific or not?
Well, we have to identify the word specific here means. Is it specific to an antigenic exposure or specific to the disease? So if it's specific to the disease, we usually try to compare the expansion of these clonotypes in different diseases. Like we take the PSC, I mean, I haven't shown it here, but we take also other autoimmune liver disease like BBC or or autoimmune hepatitis, and we try to look how the expansion looks like in there. And our assumption was if it's expanded only in that particular disease, it can also be detected, because if it responds to EBV, everyone is infected. But if it was only expanded in there, that this might be a PSC specific. Of course, we can't really say that's across all diseases. We can just say within the diseases where we did this kind of comparison, like the IBD, PSC, and the autoimmune liver diseases and primary biliary cholangitis, that most expansion was seen with PSC. The second part about the response to antigen, we can't be 100%. For some clonotype, we can be sure because I haven't shown this. We were asked by the reviewer. We took blood from healthy individuals. We incubated them with, we did tetramer staining with the BFRF3, with EBNA1 and BZLF1, and then we could show the same clonotype. So for subset of these clonotype that interact with EBV, we are quite sure that they are EBV, but for everyone else, this was based upon statistical mapping, and it's also only based on the beta, not with the alpha. So we have to take this with a little bit of a grain of salt.
Great. Sharon says, thanks for the great presentation. Really beautiful work. What are the dominant HLAs that present EBV-derived antigens?
Sorry, can you just repeat the last part?
What are the dominant HLAs that present EBV-derived antigens?
The problem is 95% of the population is infected with EBV, assuming that you reach the age of 25 to 40. With children, it's a different story. So that means that most of us has been infected with EBV, and we are also able to mount an effective immune response. We can control it, and for the majority of the population, the EBV maintain in latency stage. It just in some individuals, we don't know what happens, that this kind of EBV start to get reactivated again. So I would say there is not a specific HLA that has been linked or associated to, everyone get infected and the HLA being presented, there might be different antigen or different part of different epitopes being presented by different HLA allele. The association can be different, but I think every one of us, of course, it's really interesting to look at the 5% who are EBV negative, but the 95% of us have been exposed to EBV and have been presenting Epstein-Barr virus antigen. So maybe we don't know this in database, but if we understand, from an immunological perspective, I think everyone that infected with EBV and can control the viral titer has generated a T-cell response to it. So it has been presented by an HLA.
Great. So the other two questions, I will save it for the end as they're more open-ended. So first, I will let Stan do the demo.
Yeah, I wanted, there is one question which Dmitry wanted to ask for a while. So
sure.
Yeah, thank you very much. Very exciting presentation. I have a question about the plot that you showed where you compared differential expansion between carriers and non-carriers of HLA and within the control group and
I can also share the slides.
Okay, yeah, and PSC. So my question is, what do you think why in control group, within the HLA-B8 carriers, the expansion of your clonotypes is higher.
This one, right?
Yeah.
I mean, I can assure you that this figure took us a lot of time until we can, like, we got to digest. And I will tell you, like, how I think about it. The clonotype we identify are conventional, and they are restricted toward these alleles. So they would recognize peptide presented by the HLA-B0801. They will be present in carrier because the carrier also have been infected with Epstein-Barr virus. They have been infected with EBV. They presented EBV and generated the T-cell response toward EBV.
Okay.
So they are present in there. It's just like that in PSC, there is an EBV reactivation, so there is an expansion of these cells. But the clonotype you'll be able to get in healthy control and also in PSC individuals, because it's not like it's the same antigenic exposure happening. It's just there is an EBV reactivation happening in cases relative to control. And as a result, because most of these clonotypes are specific to the like B0801 or the DRB0301, there will be more of these clonotypes in carrier relative to the non-carrier.
Makes sense. Makes sense. So you think this is the still like antivirus T cells rather than maybe just, I don't know, anti something. I mean, environmentally, just.
I mean, what we think about is like, the Epstein-Barr virus is in there. It's one factor. It's like, it's present. Of course, there might be other antigens. It can be the microbiome. It can be a lot of other things. What we can annotate or what we can predict now is like really the top of the iceberg. And what we are currently doing is try to expand this toward like other like bacterial, microbial, maybe there is also a second co-infection with other virus. So there is a lot of things that can happen, but the easiest signal for us, or like the lowest hanging fruit in terms to say, was the Epstein-Barr virus. And that's what we managed to like, show from the T and the B cells. But of course, it's not it yet. So there is more work to be done in the future.
Yeah, it definitely makes sense. Yeah, like that would be nice if there was a way how to subtract kind of this from like all the effects to maybe isolate something even more specific. But with EBV, yeah, then you would be subtracting EBV. So yes, that's not that trivial. Thank you.
My pleasure.
So yeah, my turn. Yeah. Well, thank you very much again, Hisham. That's really impressive study, and it's a big study. So now my turn. We have a couple more questions, which we will answer at the end, and maybe my part will answer some of them. So actually, let me start sharing my screen. So yeah, my part is about the analysis. So specifically here in this more interactive section, I wanted to show you the software that we developed, and we were developing for some pretty long period, and now it's available. It's called Platforma. Historically, our first software is MyTR, which I think many of the participants use for TCR and BCR analysis. And now we have a bigger software. It's called Platforma, where you can go much deeper into the analysis and get valuable insights from the data. So for this interactive session, I took the data that Hisham generated in the study and just played around with the data to show what we can do, how we can reproduce some of the results or enrich the results of the study with new findings using Platforma and blocks available on Platforma. One note here is that for this interactive session, I took just a subset of samples from the study. So first of all, we used the samples that were analyzed, that were processed with adaptive biotechnologies. Yeah, there are 1,400 patients. So we run this whole analysis on our big server in data center, but it is so big then it's not feasible to show like interactively live on this webinar. So I took a really small subset of samples, just 20 samples from your study, 10 from a control and 10 from PSC condition. And even from this 20 samples, there are 3,600,000 of unique TCRs in this subset of samples. So now let me go through some interactive analysis with Platforma software. Well, first of all, Platforma, as you see, has an interactive graphical user interface where you can do the analysis from upstream to downstream. Yeah, and let me create just some example project to show you how this analysis can be done and how you can navigate from the upstream to downstream. Once I show something here, I will switch to the data that we analyzed from the study. So every project starts with the samples and data block, which is like the universal place where you need to import the data, the raw data or pre-processed data that you have in the study. And you are doing this by creating a new data set where you can bring the FASTQ files or TSV files. In the case of adaptive immune-seq, we have to import TSV files that are provided by Adaptive, or if you have FASTQ files, you import FASTQ files. And there are two ways how you can do this. You can import files that are located on your laptop, or if you have Platforma connected to your let's say data storage, to your HPC cluster, your university, or whatever you have where you store the FASTQ files or pre-processed files, then you can access to that data and import all the data to create a data set. Here, for example, I imported some FASTQ files. Yeah, and as you see, the platform automatically identifies read one, read two structure from the reads. You can let's say, assign the name to the data set. In this case, in case of this example, this has ERSA from RNA, create the data set, and you have your samples, you have FASTQ files, you can import whatever metadata you have. And this is very important because to do the downstream analysis, obviously, you need to provide the context for the downstream analysis. And let's say here, I have an example of metadata for this study with just Excel file where you have your sample names and then whatever metadata conditions you have. So, for example, in the study that we are discussing, we have basically one metadata column. It's condition that I imported for the downstream analysis. It's condition meaning that for each patient, for each donor, we know either this is a control or PSC. Yeah, but if you have more metadata columns, then no problem. You can import whatever you have. So once you set up the samples and data, the second part, the next step of any analysis in Platforma, well, in any analysis in real life, is to get the clonotypes. If you work with the pre-processed data, then it's one story. You use import VDJ data, and I will show once I switch to the Hisham's project. Or if you have FASTQ files and you need to extract the clonotypes, then you can use MyXR clonotyping. And by the way, here you see one important thing. I click Add Block, and you see a registry or gallery of applications or blocks that we have on the platform for various biological applications. So we have apps here, or we call them blocks for upstream analysis, like MyTR or importing G data. We have blocks for just since we are talking about T cells here, we have blocks for various repertoire analysis, like diversity, gene usage, other things for clustering of clonotypes, and I will show the things for HLA annotation, actually, and that's what I will be showing. And the whole idea of the platform is that you start building your analysis from this upstream, from samples and data, where you import your data sets, to the downstream, starting from the clonotyping or importing the data, and then all of the downstream analysis things. Now let me go to the project which I quickly processed actually before our webinar using a subset of samples published in the data. So this is all TCR beta, and this is all pre-processed TSV files from adaptive immune-seq. So as the next step actually to import this data and make it available for the downstream analysis, we use import VDJ data block, where you have settings, you specify like the format of the data, and you can import immune-seq data, you can import MyTR data, you can import any custom TSV or CSV tables with the clonotypes to make them available for the downstream analysis. And so once it is done, and this is the idea of the platform, you have clonotypes that are generated by this import VDJ data block, and they are now available for the downstream analysis. And in the downstream analysis, we can play around with the data in various ways. So we can do some classical things like clonality or diversity analysis. For example, here I added a repertoire diversity block, just for example, to look at the data, I added repertoire diversity block, which is just the app, which allows to calculate various diversity metrics or clonality metrics. Yeah, by default, there are some like observed diversity, Shannon winner. We can also add additional metrics. We can specify here, and this is actually what you see here is the default, so you don't need to play around here. You just click the run button and we'll start the analysis with the defaults, which are already good. One important thing here that for the repertoire analysis and for comparison of samples and diversity of samples, you need to do them down sampling or normalization, because obviously in different samples you have different amount of cells, of RNA, types of sequences. So for proper comparison, you need to downsample. And that's what we have here. By default, it's random sampling to normalize the data set to the same abundance. And in the case of adaptive immune-seq, the abundance is actually measured in terms of unique molecular identifiers. So it's UMI data and the abundance is in UMI. And now, for example, we go to the diversity graph in the repertoire diversity, where we can do different things and different visualizations of the data. I'm just, again, before I go to a deeper biology, just playing around with the software. And by default, you will see something like this, where for every sample you see, in this case, it is normalized Shannon winner, what is showing on the y-axis by default. Yeah, and for every sample, you have the normalized Shannon winner index. Yeah, and now, since we have metadata defined in our project, all of the data, and actually that's a key feature of the platform, allowing to do many different things, that all of the data that exists in the upstream blocks become available to the downstream blocks automatically. And you don't need to think how to integrate this data, bring on the plots, connect, like, I don't know, clonotypes with samples, et cetera, et cetera. That's all automated here. So that's why in the metadata, we have condition. That's a metadata column from this, from samples and data. And I can bring this condition as a primary grouping to see and change the type of the visualization to, for example, box plot, to see how the diversity of the repertoire differs across the groups. Yeah. In this case, and in the case of this subsampled 22 samples, it's not like super interesting. Yeah, but in general, this gives an idea of what you can do with the platform. You can also bring like p-values on the plot, and here there is no actually p-value. Do group comparisons, peer-like comparisons, change the type of visualization that you have. I don't know, change the colors, change the order of groups, and make these visualizations really beautiful and publication ready in many cases. So the same way you can do, let's say, for example, gene usage analysis. And on the gene usage analysis, again, this is the basic thing which people look when they do the repertoire sequencing. You can use the same condition to, let's say, annotate our samples to see the difference between the control and PSC samples and what the V and J gene usage for among those groups, etc., etc. So all this exploratory tools allows you to do like the basic repertoire analysis. And now let's go deeper to some of the findings in this study. So one of the features of the study is association of the HLA type with the condition with the PC versus control. And now let's go to this. So one of the blocks that we have in the platform is ImmuneWatch Detect. That's actually a block allowing to predict TCR specificity based on the machine learning algorithm, and not only the TCR specificity, but also an HLA associated with this TCR. So we have this block added here to process this data, and now just to show you, the clonotype browser is the block allowing you to explore, like on the tabular view, the whole repertoire that you have in the project. For example, here in the per sample tab, we switch between samples. On the overlap, we have all of the clonotypes across all of the samples that we have, with all of the properties like the number of UMIs in different samples, etc. So here we have 3,600,000 of the clonotypes, so the data takes time to load. But in general, let me go here on the per sample, for example, you already see that in this, once we run the detect tool, for every clonotype, we have associated HLA type predicted by detect. And also, well, that's what we are going to use downstream, and also the antigen. Actually, we also have predicted antigen for every clonotype coming from the detect. So let me reload because it's loading from the server. So now one of the really powerful tools that we have to explore further the repertoire is called clonotype space. And let me go to the clonotype space right away. This is the block allowing to visualize all of the TCRs that you have in the whole project on one UMAP space. So this UMAP has 3,600,000 of clonotypes, so it takes time to load. Let me refresh it. 1, 2, 1, 2, 3. 1, 2, 1, 2, 3. So it's loading, it's loading, it's loading, it's loading. It is loading. Yes, now it is loaded. So you have, yeah, well, it's 3,600,000 of dots here on the UMAP. Yeah, so it takes time to load. And actually, when you do the when you run the project on the whole data set, it has 3 billion of unique TCRs. So it will take more time. And for that, you actually need pretty powerful laptop just to visualize that UMAP space. So there are certain limitations. When you do population studies, there are certain requirements for the hardware. So here we have a UMAP, and this is the idea of the UMAP is that 2 dots are located closer to each other if they share similar properties. Yeah. So one of the things that we just, for example, can do on this UMAP is to color by the G gene. I don't know. I just picked it as an example because for G genes, that's what I know. You have a nice clusterization. This global structure of this UMAP is dominated by the G gene because the similarity of the TCRs is calculated based on the protein sequence of the in this case CDR3 region, and the G gene is the thing which allows to separate them more nicely on the UMAP. Yeah, and the second thing that I mentioned, that all of the data that we generated in upstream becomes available downstream. And here in the clonotype browser, I did one thing. This is actually a very powerful instrument allowing you to select and annotate different subsets of clones. So we have, from the detect block, we have HLA predicted here. That's just like a very well-known tool to predict HLA and TCR specificity. And I used this as an annotation to filter only those clones, only those T-cell receptors across all of the study that are specific to the HLA-B08. I also filtered by the score, with the prediction score. So detect is a machine learning tool. It's algorithm using trained models, etc., and it provides a score. And actually, that's some recommendation to put some threshold on the score to just have a nice confidence on the results. And now this information is available for me downstream, on the highlight. I highlighted only those TCRs, only those clonotypes that are annotated by detect as HLA-B08, and you can play around here. You can zoom in. You can select a subset of clonotypes with lasso, compare them on the multiple sequence alignment to see what are the motifs here, etc., etc. And you can apply further filters. Let me clear this. You can apply further filters in the data mapping to navigate to some more specific sequences that you have. So this just gives you an idea. All of the data that we have here, we can use number of UMIs supporting the clone types as a size of our dots, or we can filter to a specific species. Or let me actually, here I did one more thing just to play around with the data. In your study, you have a list of clonotypes that are PC associated. So we can actually import this data and match those PC-associated T cells with the whole base of our TCRs in the project. There are some...
Sorry, and we did the same. Yeah, you can set up a certain level of fuzzy search.
Yeah.
So yeah, you can expand around the clonotypes of interest.
Yeah, that's actually what we did here in the matching parameters. So to match the PSC-associated clonotypes, we use the alignment score for the match. We use like Blossom, or we can use exact match. The threshold that I put here on the subset of the data is 90%, and the coverage threshold is 95%, meaning that the length of the TCR, we allow to vary the length when the matching, but the length difference should be within the 95%. Yeah. So we run it, and let's say here we can use this PSC associated clonotypes as a filter, yeah, to include only PSC associated clonotypes that matched the PSC associated clones. Once it loads, again, it runs like this join on 3.6 million of clonotypes. You see a filtered subspace of the clonotypes that only match them. And the highlighted ones are the ones that are specific to the HLA that we selected here in the highlight, for example, again. And then you can play around and let's say use a specific antigen predicted by the tech. Again, this is one of the tools available here as a label, you know, to label them, all of the dots that we have here, to label with the predicted TCR specificity. Or actually, if you zoom in or if you play with the table, the rest, yeah, you see Epstein-Barr, you see Epstein-Barr, yeah, Epstein-Barr, yeah. So we already see this just with clicks. We see nice patterns here, which is actually nice. So, and I'm finishing. So the last point where I wanted to show you what we also have on the platform, how you can play with the data and find your insights, is that like for this exploratory studies, actually the end point of many exploratory studies is the graph maker. That's the block we have to do a comprehensive visualizations of whatever data you have generated in the upstream to your project. And here specifically, I built as an example, just one plot. On this one plot, I used clonotypes that we annotated with HLA. We used the detect output that predicted us the HLA. We used this annotation, and we used here only the clonotypes that are annotated with this HLA type. On the y-axis, we are showing the fraction, the abundance of those clonotypes in the samples where they're present. And as a primary grouping, we use the condition that we have. And with this plot, Dmitry, you comment better what this plot shows.
Yeah. Basically, this is one of the papers here that you cite that this HLA association. It kind of shows this HLA association, but on the level of repertoire, just measured without any genetic, any genetic testing, any genetic information. So basically, that's it.
That's it. Yeah. So we have this as some like thing that we clicked around just before the webinar to show that you can do things like this. So that's what I wanted to share. It's with this level of depth in your study and the scale of the study, it's pretty complicated to reproduce all of the results in 20-minute webinar, but it gives you an idea of what you can do with the software and how you can, in your future studies, navigate in Platforma to find those things. So thank you very much. I finished. Monica.
Thanks so much, Stan. So Hisham, that was a really good summary of your massive study. And thank you, Stan, for showing us how to do something similar with a no-code bioinformatics platform. So for Platforma, just like MyXCR, it is free for academia. And then I will send out a link with the recording of today's meeting so everyone can know how to access it themselves. So let us move over to the questions. So we had a question from Sam, just very generally, Hisham. Have you considered looking at Burkitt's lymphoma?
Yes, I mean, Burkitt lymphoma is a very interesting disease because I mean we know that the EBV is in there. I mean, I would love to do this, but we don't have basically access to biological material to do this. But it would be interesting. So I mean, we can talk about this if you have the sample. So that's a fun project to work on.
Yes, Sam, if you have samples, I'll connect you with Hisham. Okay, and then one other question from Darren. Do you have any thoughts on tissue specificity of autoimmune reaction in PSC through EBV infection?
That's a great question, and that's where we are basically now heading now, because now we know that there is there is this kind of EBV reactivation, there is this signals, but we don't know exactly how this can lead to the disease. We don't know if also if the EBV is, we know, from the what is called from the TriNet X, from the population level scale of medical record, that getting an infection mononucleosis is associated with the risk of developing PSC, or they are both associated. However, how this mechanistically happened, we can speculate, but I can't really give you an exact mechanism because we don't have the data to support it. It can be that basically there is a chronic T cells activation, and this chronic T cells activation is not able to clear the infection, and then we have the exhaustion marker, or it can be because of there is something that's going wrong with the infection. There might be some kind of EBV reactivation happening, unresolved infection. There is a lot of things that we can speculate on, but to basically we need to basically wait until the data is generated, and then we can have a data supported framework for it.